Hello, everyone, and welcome to Visions 2022. My name is Claire Gelfman, and I'm the Chief at the Foundation Fighting Blindness. We are very excited to have you all here today. Background about our organization. Our urgent mission here at the Foundation is to drive the research that will provide prevention, treatments, and cures for those affected by retinitis pigmentosa, macular degeneration, Usher syndrome, and the entire spectrum of retinal degenerative diseases. And today, we are the leading private funder in the world of retinal disease research, we just celebrated our 50th anniversary. Well, here at Visions 2022, you're going to have the opportunity to join some scientific sessions. And the purpose of the next 20 minutes or so is to give you some background so you can enter those sessions with a little bit of information that will allow you to really maximize the information being presented. And those topics include gene and genetic technologies, optogenetics, stem cells and retinal disease research, genetic testing and counseling, and then we'll end with an introduction to our Retinal Degeneration Fund, or RD Fund, which is a venture philanthropic arm of the Foundation. Well, we all come to this conference with different levels of background. So what I'd like to do is spend a couple of slides, a little bit of time, reviewing some ocular anatomy and physiology. So this slide shows a schematic of the human eye in cross-section. So on the left-hand side of the slide, if light were entering your eye, it would enter right through the pupil, and that light would become focused by the cornea and the lens, and the light would then land on this back outside wall of the eye called the retina, which is actually made up of several different layers. And we're going to be talking today a lot about photoreceptors, and it's those photoreceptors whose main job in life is to capture the light coming in through the pupil and then transmit that light to a message that our brain can interpret so we can actually know what it is in front of us, the image in front of us. And the conduit of that message is our optic nerve, shown here in the middle of the slide. Now, photoreceptors come in two different flavors, rods and cones. And it's rods that allow us to see at night under low light conditions. So there's much less detail that our rods give us, but they're much more light sensitive, allowing for that night vision. Cones, on the other hand, allow us to see under brighter light conditions and give us our perception to fine detail, as well as our color perception. Cones are concentrated in the foveal region of the macula, and there are about 6 million cones in that region compared to 120 million rods. So rods are found more in our peripheral retina, where cones are more in our central retina. So let's talk about the central dogma of biology, which simply states that in every cell in our body, we have molecules of DNA, and those molecules of DNA will get transcribed into an RNA molecule to then go on to be translated into a protein. And it's the proteins that keep us alive and well. So if you have a mutation or some change in your DNA and you can't make a functional protein, then that's when disease will ensue. And that's exactly what happens in a lot of different ocular diseases. You may have heard of Luxturna, the only gene therapy approval for a retinal disease. And it's giving back a good copy of a gene at the level of the DNA called RPE65, so the right protein can be made. I would be remiss if I did not also mention that the COVID vaccine is an RNA type of vaccine. So the central dogma fits across all aspects of our life when it comes to areas of therapeutic intervention. There are several different types of ocular gene therapy. I mentioned Luxturna, and it falls under the category of a viral-based gene therapy, and it uses a special virus known as an adeno-associated virus. And this is currently the most promising vehicle for gene delivery to the retina. Now, we've also heard a lot about gene editing, especially CRISPR, and this is a little bit different. With the AAV delivery of a gene, you're giving the entire gene back to the cell type of interest. CRISPR gene editing is where you go in and edit one specific base, one specific area of the gene. And in fact, that method is being leveraged in a gene therapy program at a company known as Editas that's using CRISPR to bring in to gene edit a special gene known as CEP290 for individuals who are affected by a special type of Leber's congenital amaurosis type 10. On the right-hand side of this slide, I have a picture of both Jennifer Doudna and Emmanuel Charpentier, who won the 2020 Nobel Prize in Chemistry for their discovery of CRISPR. So we're going to see a lot more about different types of gene editing in the future as it relates to ocular disease. So I mentioned that adeno-associated viruses are currently the most promising vehicle for gene delivery to the retina. And in order to understand an AAV, there's really two components that are important for today's discussion. One is the capsid, which is this protein-based shell that really has the instructions upon delivery where this AAV should go. What's the target cell type? And then inside that capsid, inside that protein encasing, if you will, is the DNA. And it's that DNA, if we think back to that central dogma, it's the DNA that gets expressed to make a therapeutic protein. Now, there are certain size limitations to the DNA that can fit inside one of these AAVs. So part of where the field is going, in addition to those where the correct DNA is able to be utilized, we're also looking at a lot of new technologies that allow for larger genes to also fit into an adeno-associated virus as a means of gene therapy delivery for ocular diseases. So what are some of the facts? We know that mutations in over 300 genes can cause inherited retinal diseases. And in the case of retinitis pigmentosa, there are about 70 of them. So RP has an incidence of about 1 in 3,000 worldwide, so that's about 2 million people. So when we think about ways to treat retinitis pigmentosa, since frequently we know the causative gene, the therapy is simply to augment with a good copy of that gene to provide a functional copy of the DNA so that the right protein is therefore made. Gene editing, as I mentioned a moment ago, is similar to that, except it allows for specific editing at a specific place in the gene. And we'll also hear over the next couple of days about RNA-based therapies that utilize antisense oligonucleotides as a means of therapy. So lingering questions lead to alternative approaches. So what if your gene has yet to be identified? You go through genetic testing, or maybe your gene's been identified, but there's no one working on it. I want to introduce the concept of mutation-agnostic approaches because you're going to hear a lot about those in the next couple days as well. And the advantage of these approaches that are not necessarily dependent upon knowing your causative gene would result in treating more IRD-affected individuals with a single therapeutic. And these mutation-agnostic approaches for the treatment of IRDs are based on the pathology, so knowing what's causing photoreceptor degeneration. You can have 10 people in a room with a different causative gene, but the vision loss is due to a degenerating photoreceptor. So let's talk for a moment about how we can focus treatments on a degenerating photoreceptor. And there are lots of therapeutic approaches that are being leveraged. The one thing we know is that oxidative stress causes vision loss. Oxidative stress really compromises vision. So no surprise then that there are antioxidant approaches. So these are not gene therapy based necessarily, but antioxidant approaches for the treatment of inherited retinal diseases. And we're going to hear about some of those over the next couple of days. There are also visual cycle modulators, for example, from the case of Stargardt, that can reduce the buildup of these toxic intermediates that can compromise vision. We're also going to hear about complement inhibitors for the treatment of the dry form of age-related macular degeneration. Now, there are some mutation-agnostic approaches that do not rely on knowing your causative gene, but still utilize a gene therapy to bring in a special protein. And we're going to hear about two different techniques that fall in that category, one being neuroprotection from a company called Sparing Vision. And we're also going to hear a lot about optogenetics. And I want to spend a moment talking about optogenetics because it's getting a lot of attention, not only in its preclinical development, but also in human clinical trials. So simply stated, optogenetics is a technique that involves using gene therapy to transfer a gene that encodes a light-sensing protein, which can turn a cell that normally would not sense light into one that can. So remember I mentioned earlier that photoreceptors have that important job of capturing light and then transmitting that light to a message that our brain can interpret. And that really defines, very simply, functional vision. But what if your photoreceptors are degenerating? What optogenetics does is it gives that light sensing capability to a part of your retina that is not degenerating. And it has the potential to help people who have lost all function of their photoreceptors, because unlike gene therapy, it does not require any semblance of a functional photoreceptor cell. There are several companies that are working on optogenetics as potential therapy, so we're going to hear a lot about those over the next couple of days. We're also going to hear about stem cells in the retina as a therapeutic approach. And simply stated, stem cells in retinal disease allow for taking healthy photoreceptors that are derived from stem cells and then transplant those into a patient's retina to replace the lost photoreceptors. So how this works is that over time, the transplanted photoreceptors are left to mature, making connections with the recipient's remaining retina, the idea that then vision will be restored. And current efforts are focusing on cone photoreceptor-rich regions of the macula, which is the central zone of the human retina that will hopefully enable high acuity vision for tasks such as facial recognition and reading. I mentioned before that Foundation Fighting Blindness is unique in that we have a venture philanthropic arm that we call the RD Fund. And this allows us to invest in companies that are developing therapies for both inherited retinal diseases and dry AMD. And the financial returns from these investments come back to the foundation to further our mission. And this helps new companies advance their treatments into early stage trials, as well as helping these companies attract investments from outside capital. The RD Fund is a subsidiary to the Foundation Fighting Blindness and is governed by a separate board of directors. So I've been talking a little bit about age-related macular degeneration, and I wanted to spend a couple of minutes because AMD is a leading cause of vision loss in individuals over the age of 60. And it's due to damage of the macula, this light-sensing part of the eye that's rich in cones, controlling our sharp central vision. So when you have AMD, it's your central vision that goes first, but then it can extend outside into the periphery, eventually leading to full blindness. It comes in two different forms, and the wet form is the blinding form, and it's due to the vision loss from wet AMD is due to this abnormal blood vessel proliferation and leakage due to a growth factor known as vascular endothelial growth factor. Now, the good news, if you will, is that if you get diagnosed with wet AMD, there are treatments that are working and being used successfully in the clinic. And these treatments are targeting these VEGF molecules. So as I mentioned, there is a wet form and a dry form of AMD, and it's the wet form that constitutes for about 10 to 15% of all patients that get diagnosed. The remaining 85% have the dry form, dry AMD, and you can actually have dry AMD initially and not even know it, get the diagnosis when you go for a routine visit to your ophthalmologist. But if the dry form progresses, it can get to what's called late stage geographic atrophy. And the cells that are atrophying are it's a single layer of epithelial cells just beyond the retina. And that can cause vision loss if dry AMD progresses to geographic atrophy. Well, unlike inherited retinal diseases we were talking about, AMD is not due to a single causative gene. In fact, it's what we call polygenic. Many genes, when mutated, can cause the development of age-related macular degeneration. And it's actually more complex than that. There are environmental factors that increase one's propensity to developing AMD, multifactorial in that regard. There are a lot of approaches, though, for treatments that instead of being based on the genetics per se, they're based on the pathology. So one thing that we know, what we've learned, is that there are pathways that go awry during different types of macular degeneration. And one of those pathways is called complement. And the role of complement in all the cells in our body is simply stated to get rid of things that don't belong, pathogens, for example. And what we've learned is that the complement pathway is on overdrive in individuals with wet and dry AMD. And in fact, complement pathway modulators in dry AMD are being evaluated right now in human clinical trials, and we expect to see results in phase 3 studies in the next 6 to 12 months. There are also cell therapies that are being leveraged for the treatment of AMD, where the cellular environment is restored. We also here at the Foundation Fighting Blindness have a program called My Retina Tracker, and this is a genetic testing program. So I mentioned before about the importance of knowing your causative gene, if possible, because these are mainly monogenic conditions. And the importance of testing can learn what the genetic basis for your clinical diagnosis. It can also confirm or sometimes even change the diagnosis based on the results of that genetic testing. And it can really help provide a more accurate prognosis. And importantly, for your family, it can confirm the inheritance pattern so you'll know the risk of other family members. And it can also help you qualify for human clinical trials if you do know your specific, the genetic cause. One of the things that impressed me when I first came to Foundation Fighting Blindness about a year ago was the fact that if an individual gets a diagnosis from their ophthalmologist for an inherited retinal disease, we offer free genetic testing from our partner at Blueprint Genetics. Now that genetic test, once you have those results, can be quite daunting. So we also offer free genetic counseling through our partners at Inform DNA. And all that information then gets captured in our registry called My Retina Tracker Registry, that as of the end of May, at the end of last month, we had over 21,000 members in our registry, about half of which were tested through our genetic testing program. And you're going to get to learn a lot more about genetic testing and counseling and My Retina Tracker at the plenary session on Friday, the science plenary session. So that is not to be missed. Well, in addition to the science programs that I just mentioned, we have a lot more in store for you over the next couple of days. We're going to hear from blind athletes who are breaking barriers in their specific sports. We're going to hear about how low vision occupational therapy can help. We're also going to hear about the impacts on our mental health, particularly anxiety and depression that can come sometimes with that diagnosis, not only for patients, but also for those in a helper role. We're also going to hear about ways to increase mobility and ways to use technology to really live your best life with a diagnosis. We're also going to hear about medical ethics and clinical trial participation. I'm very excited about a couple different keynote sessions. So at lunch on Friday, Chad Foster is going to talk about his journey. If you haven't already read Chad's book called Blind Ambition, How to Go from Victim to Visionary, I highly recommend it. It's a very inspiring book. Chad was the first blind person to graduate from the Harvard Business School program for leadership development. He currently works at Red Hat. He's been blind since he was in college due to retinitis pigmentosa and is a very, very inspirational speaker. And speaking of inspirational speakers, our closing session keynote, we'll hear from Dr. Marsha Link, who is a clinical psychologist with a very diverse background that expands both the ophthalmology sector as well as being an executive coach and educator. And she served as president for 3 years of Ophthalmic World Leaders and is an amazing speaker. So stay tuned for that closing session as well. And all of which we're presenting to you over the next couple of days here at Visions in Orlando would not be possible without the very the generosity of all of our sponsors that I've included on this slide. And that includes Janssen, Genentech, Spark, AGTC, Iveric Bio, Apellis, Nacuity Pharmaceuticals, Regenix Bio, and Two Blind Brothers. So we're so excited to have you here in Orlando for Visions 2022. Enjoy the next couple of days, and if you have any questions, feel free to go to our website at www.fightingblindness.org. That's www.fightingblindness.org. And again, welcome to Visions.